One of the most important and very commonly used chromatography technique in molecular biology, or to say for other analytical purposes, is a gas chromatography. Now, gas chromatography is a chromatographic technique that can be used to separate volatile organic compounds, as the name implied, that the organic compound which are volatile, it can be used to separate those molecules. Now, in the gas chromatography, it consists of a flowing mobile phase, as in chromatography. In all the chromatographies, we have a mobile phase and another phase, and an injection port where we inject the sample, a separation column. Of course, that should be called a stationary phase, and this whole thing is put in an oven for a temperature, and then there are detectors which detect the material which comes out of the column and indicate this. For example, in this diagram, the component of a gas chromatogram that has been depicted, and as shown, there is a column which is in the oven, put in the oven, then there is an analysis board, sample outcome, and then there are detector. Those detector, the reading, it goes to the computer where it is indicated that different compound, whatever, where they are present. Now, the organic compounds, they are separated due to the differences in their partitioning behavior between the mobile gas phase and the stationary phase which is present in the column. So, in this case, what happened that the partitioning behavior of various compounds is a major factor, identification factor for those molecules. Now, the partition coefficient, they are inversely proportional to the volatility of the analyte, so that the most volatile elute first. Definitely, the compounds which are most volatile, they will come out of the column first, and then the other compound, they will follow. The stationary phase consists of a high boiling point liquid material such as silicone grease. That is the stationary phase in the column, and there is an optimum flow rate of the mobile gas phase for maximum column efficiency. That is, the flow rate is also should be optimized for getting the maximum column efficiency which is there. Now, gas chromatography is widely used for the qualitative and both quantitative analysis of a large number of low polarity compounds. That is its characteristic, that it is used for qualitative as well as for quantitative purposes for a low polar compound, because it has got very high sensitivity. It is very sensitive technique, it is very reproducible technique, and it has got a speed of resolution. That is, it can resolute, it can differentiate between different compounds which are present there in the sample very effectively. Now, analytically, it is a very powerful technique when compared to the mass spectrophometry. So what happened that the gas chromatography can be used jointly with the mass spectrometry, which further differentiate, which different classified or specifies the compound which are there, and the column which are used in the gas chromatography, they are, number one is the packed conventional column. That is different type of columns which are available commercially. They are packed, or they are capillary open tubular column. That is the capillary column, very large. They are also available. Now, several type of detectors. Now, one, these are the column. Then another essential part is the detector. Now, the detectors are in use in conjunction with the gas chromatography. They are the flame ionization detector, known as FID, nitrogen phosphorus detector, NPD, and electron capture detector, ECD. These are the main types of the detector which are used with that. Now, flame photometric detector, they are the rapid scanning Fourier transform infrared detector. They are also used with this. And then there is a mass spectrometer detector. That is, when we use in conjunction with the mass spectrometer, we can use mass spectrometer detector where we can analyze a compound further, precisely and completely with that. Now, in this diagram, the gas chromatography flame ionization detector is indicated, that how it operates. There is an air supply, there is a hydrogen supply which is there, and the flame is there, and then there is a detector which detects the compound and gives the amplification to a computer. Now, advantages of this technique is that it requires only very small sample with little preparation, because the volatile compounds, they are detected there. So we need a very small sample in microliters. Then it is good at separating complex mixture into compound. We can use it also for separating the different compounds which are present in a mixture. Then the results are typically obtained. The results, they are rapidly obtained in this case, and usually it takes within 1 to 100 minutes, that within 1 to almost 1.5 hours, we can use this technique. Now, it is, as indicated earlier, it is extremely precise technique. It is extremely precise technique for using in the analysis, and only instrument with the sensitivity to detect volatile organic mixture of low concentration, we can use the gas chromatography technique. And as said earlier, the gas chromatography technique is used conventionally and repeatedly in the case of molecular biology due to the properties or its characteristics as indicated earlier in this lecture.